All right, so yes. Okay, so here we're going to talk now about the synthesis of our pyrimidines. Now, in the synthesis of pyrimidine nucleotides, the base is synthesized first and then it is attached to the ribose. So in this case, you have base first, then attached to ribose or sugar. Now, your initial reaction is going to be analogous to the urea cycle, except it uses glutamine rather than ammonia. And also, it's going to occur in the cytosol. So remember, in the urea cycle, we had CPS1. Well, now we have CPS2, carbonoyl phosphate synthetase 2. And this is in the cytosol, not in the mitochondria. Okay. And so here we have, again, we're going to have glutamine and a carbon source from carbon dioxide, and this takes 2 ATP to generate the carbonoyl phosphate. Okay, so now the catalyst by carbonoyl phosphate synthase 2 is the regulated step of this pathway. And you can see that it's going to be positively regulated by that activated ribose, BRPP, that we talked about. After you get carbonoyl phosphate, the next step is you get an entire aspartate molecule added to the carbonoyl phosphate, and this is by aspartate transcarbamylase. This is also going to subsequently closes to produce the ring, and this is going to be by a dehydroorotate. Oops, this closes the ring. And then you're going to form orotate. And so orotate is going to be added to our activated sugar that we talked about in our purine synthesis. And orotate will initially form UMP. Okay, and then UMP can go on to form our other nucleotides, our cytosine and our thymidine, shown there. Okay, so the pyrimidine ring is going to be made by a full aspartate molecule. It's also going to be part of the carbonoyl phosphate, which was derived from, if you remember, carbon dioxide and glutamine. So you have the N from glutamine and you have the carbon from carbon dioxide. And that's basically a pyrimidine ring. And so this is just to go over this again. So you have your aspartate and you have your carbonoyl phosphate, shown here. And this is going to generate what's called a carbonoyl aspartate. The enzyme, it will form orate or auric acid. Now, auric acid shown here has a step catalyzed by your aurate phosphoribosal transferase. And this is an important enzyme because there are deficiencies associated with this enzyme, as shown by this line here. Oops, wrong one. And so the aurate phospholipase transferase transfers from the ribose 5-phosphate from the PRPP, which is that activated ribose, to orate. And this is what's going to make OMP. So you're bringing in, oops, I have the wrong one, sorry. You're bringing in your activated ribose group, this PRPP, and then you're forming that first OMP. Now, OMP gets decarboxylated by the orotylylic acid dehydrogenase, and this will form uridine monophosphate, and that's shown here. And so this line here is showing what is blocked during hereditary uric aciduria. Now, hereditary uric aciduria, we'll talk about that here. So this is basically excess excretion of uric acid in the urine. And this is associated with anemia, mental physical disabilities, you have a decrease of pyrimidine synthesis. And so you get a similar thing that was associated with the vitamin B12, folic acid. You're going to get a megablastic anemia, larger cells, odd sizes. You also get a decrease in your nucleotide lipid cofactors, which are needed for synthesizing your red blood cell membranes. And so that's going to be associated also with this megaloblastic anemia. Now, once you have your UTP formed, we had our UMP, which is going to go to UDP, which is going to go to UTP. And once that's formed, you have CTP synthase. And this is going to be with glutamine plus an ATP. You are going to get CTP and glutamate. Plus one of your inorganic phosphates. And so this is how UTP goes to CTP. Now, one thing that has to happen here also is you have always on your pyrimidines, you have your ribose added. And so you need to convert your ribose to your deoxyribose. And this is going to be catalyzed by ribonucleotide reductase, shown here. And ribonuclease reductase requires a protein called thyroredoxin, shown here. So your regulation is going from ribose to deoxyribose is at the ribonucleotide reductase. And it is complicated, or it's quite complex. They have two allosteric sites. So this is your ribonucleotide reductase, and it has your regulatory sites. It's allosteric regulation, and so what that means is, of course, that you have sites separate from the substrate sites. So you have your substrate specificity site, and so in this case you're bringing in a ribose, obviously, and then you have your regulation sites. So you have allosteric effectors, and then you also have your substrates. So your allosteric effectors are going to be ATP, deoxy ATP, deoxy GTP, etc. You also have your substrates, which can be ADP, CDP, UDP, GDP, etc. Now, one of the other things about this, going back to thyrodoxin, thyroredoxin is going to require NADPH for a reducing agent. and you're going to have thioredoxin reductase, which is going to take a thioredoxin with two sulfur groups and make a thioredoxin and reducing those sulfur groups. And so ribonucleotide reductase requires that substrate in order to remove the oxygen from ribose to make deoxyribose. Now, for the regulation of this, if you have ATP bound to the active site of the enzyme, this is going to activate the enzyme. If you have DATP bound to this site, it's going to inhibit the enzyme. So right here you have, let me make this green. So ATP would be a green light, and your deoxy ATP would be the red light, and that makes sense, right? So if you have a deoxy, too much of it, you're going to want to stop making deoxyribose. And if you have a lot of ATP in the ribose form, then you want to make deoxyribose. All right, so let's go through this a little bit more. You're going to have, again, your you have your deoxy-UMP, deoxy-TMP, and the conversion of that is going to be through a synthase, thymidinolate synthase. Now, this is going to require a donation of your carbon, shown here, and this is through the N5-N10 methylene tetrahydrofolate. So your N5 methylene tetrahydrofolate will donate that carbon source to go from DUMP to DTMP. And this is going to generate a 7,8 dihydrofolate. You're going to have a reducing agent of NADPH, which is going by the dihydrofolate reductase. And this is going to generate your tetrahydrofolate. And then you have a serine hydroxymethyl transferase in which you get serine transferred. Remember, that's to generate glycine. And this is going to generate that N5-N10 methylene tetrahydrofolate, which is used to make thymidine monophosphate from uracil monophosphate. So this is tying together what we talked about with respect to our amino acid metabolism, going from serine to glycine to form our N5-N10 methylene tetrahydrofolate, which is necessary to make the base, the thymidine bases. Now, there is some salvage of pyrimidine bases. This takes 2 steps to do. The very first step is really a non-specific pyrimidine nucleoside phosphorylase, which is going to convert the pyrimidine bases to their nucleosides. So you have your bases, and then you're generating the nucleosides. And then once you get to the nucleosides, you have more specific kinases, and these are going to react with the nucleosides to form the nucleotides. So this is virtually, this conversion here is non-specific with respect to your phosphorylases. And then the next step is going to be specific kinases. And then you're going to get your nucleotides. Now there is a special case of thymidine kinase. So this is a deoxyribonucleoside kinase. This is going to be allosterically inhibited by deoxythymidine triphosphate. And so What happens is during the cell cycle, the activity of thymidine kinase will rise as the cell enters the S phase. Okay, and so this is what has been used for in labs in order to incorporate radio-labeled thymidine for isotype labeling of DNA. So basically, you radio-labeled thymidine, and you're using the thymine kinase activity in order to incorporate all these radio-labeled thymidines. And so this is going to be used to investigate or estimate rates of intracellular DNA synthesis, for example. So it's just a tool. But you might have heard of radio-labeled thymidine, for example. And the reason it's used is because of this activity of thymidine kinase during the S phase of the cell cycle. Now, the regulation of the formation of new pyrimidine synthesis is going to be regulated by that first step, the carbamoyl phosphate synthase 2. And it is inhibited by its product, UTP, and by activated sugars, ribose. Now, catabolism of pyrimidine bases is pretty straightforward. They're going to be dephosphorylated, and then the nucleosides get cleaved to produce your ribose and the free pyrimidine base, either cytosine, uracil, or thymidine. So here we have thymine, we have a dihydrouracil dehydrogenase, which uses NADPH, and you get a dihydrothymidine. And then you have a dihydropyrimidinase to generate the intermediate of your beta urididyl butyrate. And then you're going to have a urididyl propionase, which will generate a beta aminobutyrate. And then finally, aminotransferase to get a methylmalonyl semialdehyde. This is a transamination reaction. Of course, PLP is going to be involved, and you have that alpha-ketoglutarate that picks up that nitrogen. So you're going to have cytosine is going to be deaminated to form uracil. Sorry, it's an A. Then your uracil is going to be converted to carbon dioxide plus your ion and your beta alanine. And then, as I said, thymidine is going to eventually become methylmalonyl semialdehyde. All these products that are being formed All these products being formed are going to be excreted or reused. So they become carbon dioxide, hydrogen, nitrogen. Nitrogen can go to form urea. Carbon dioxide can be exhaled, etc. So we know what can happen with those. So what's important is they do not cause any problems in the body. So there is some clinical significance with this. They are the disorders that affected that OMP decarboxylase that led to the orotic aciduria. Type 1 is that orotate phosphoribosyltransferase transferase, and the OMP decarboxylase activities are both defective. In a type 2, the deficiency is only in the decarboxylase, but both have the same effect. They cause a block in DNA synthesis. You don't get your pyrimidines made. It can be treated by adding those bases downstream of those two reactions. So you can treat with uridine or cytidine, and that is going to lead to an increase of UMP. And then UMP would feed back and inhibit that CPS2 activity, so you don't get a buildup of the precursors of the pyrimidines formation. Okay, so finally, summary very quickly. We talked about in the prior lecture purine rings. These are going to require a lot of ATP and a lot of steps, 11 steps, 6 ATP. You're going to have in your purines glutamine, glycine, aspartate, nitrogen, and as well as you're going to get carbon dioxide involved in that, as well as some carbons from your tetrahydrofolate pools. Pyrimidines are going to be synthesized through the carboxyl phosphate, which is similar to what it's going to be in the cytosol, of course, not in the mitochondria during the urea cycle. You're going to use aspartate as well as your carboxyl phosphate, and then you're going to attach this, after you make this, you're going to attach this to the ribose. So that's different than the purine system, where you start with the activated ribose. You have, of course, your salvaging pathways. You have, of course, your ability to convert your nucleoside monos into tris, etc. And then you can take your ribonucleotides and convert them to deoxyribonucleotides. Thymine nucleotides come from DCDP and DUMP. Purine is going to generate uric acid. Pyrimidines are going to help to generate urea by releasing those nitrogens. The purines have to be salvaged. They can be switched back and forth between each other, as well as into different nucleotides and free bases. With association with purines are going to be more genetic deficiencies in those salvaging enzymes that cause very severe disorders. And of course, accumulation of uric acid crystals in the joints can cause gout.